Hello everyone and welcome. In this video we are going to be checking out February-March 2022, the MCQ. If you have any specific questions to check out in this variant, there are timestamps in the comments, and if you have any resources you'd like to share, obviously feel free to share those in the comments, because this is like a community where we're trying to help as many AS biology students as possible. And lastly, if you have any questions that you would like to ask, feel free to ask these again in the comments down below. Let's get started. Number 1. Which cell organelles are clearly visible when viewed through a microscope at a high power times 400? Okay, so times 400 power on light microscope. Remember, in a light microscope, the maximum possible resolution is 200 nanometers. Hence, the maximum distance between two separate points that you can visualize are going to be 200 nanometers, and we know the sizes of each of these structures. So basically we know this could be 18 nanometers, this is the smallest size, or it could be between 20 to 25 nanometers, right? The endoplasmic reticulum, now anything that is single membranes cannot be visible, just like the endoplasmic reticulum. Same thing with, like, this is non-membrane bound and its size is extremely small, but for single membrane organelles, the only distance between the two membranes is only 7 nanometers, right? So basically if you look at the structure of the endoplasmic reticulum, it's just going to be a bunch of folded structures that are going to be like 7 nanometers in thickness, right? So obviously it cannot be visualized. Centrioles, it's the same thing for centrioles. They're non-membrane bound organelles, you cannot visualize them. And lastly, chloroplasts are the only ones to be visualized, and so the answer is D. Now if you look at a cell under light microscope, magnification times 400, the only thing you'll see is the nucleus, and just basically you see it as this dark structure. Right, now let's move on. 2. Microvilli and root hairs are characteristic structures for some cell types. Which row identifies some features of microvillus and root hair? Surrounded by a cell wall, obviously only for the root hair, because microvilli they're only found in animal cells, so obviously they're not surrounded by cell walls. More than one present on a cell. Microvilli, obviously there's only one root hair in every root hair cell. Surrounded by a cell wall, as I mentioned, root hair, but more than one present, so it's A. 3. The photomicrograph is of a plant cell. The cell is 25 micrometers in width from X to Y. And what's the magnification of the photomicrograph? So for this we'll need a ruler. I'm gonna try to set this ruler like this. So this is in centimeter. Okay, so we've got from, let's say this is 0 all the way until 1, 2, 3, 4, 5, and 6 centimeters. In other words, 60 millimeters. He already gave us the cell, which is 25 micrometers in width. And so in order to calculate the magnification, we have to rely on the triangle. Let me remove the ruler now. Okay, the triangle says that it's IMA. This is image length, right, divided by actual length to get the magnification, which is M. So as I mentioned, M equals to I over A, but they have to be in the same units. So if image length was 60 millimeters, we have to convert it to micrometers, just like the actual length, which is in micrometers. So to convert from millimeters to micrometers, because micrometers is smaller, you have to multiply by 1000. In order to convert back from micrometers to millimeters, you have to divide by 1000. So what we'll do here is we're going to multiply 60 by 1000 to get 60,000 micrometers. To get the magnification, I should say 60,000 divided by, actual, which is 25, and that should give me a magnification of, 2.0 times 10 to the power of 3, or 2000. The answer is C. 4. Which eukaryotic cell structures are all contain nucleic acids? So obviously here, when it comes to nucleic acids, make sure that the answer does not contain Golgi apparatus, Golgi bodies. They have to be cancelled out. So here's first one, cancel. Centrioles do not have any nucleic acids. Centrioles, again, wrong. Chloroplast, mitochondria, cytoplasm, ribosomes, yes. Ribosomes have ribosomal RNA. Cytoplasm has tRNA, mRNA, has ribosomes, so it has ribosomal RNA. Mitochondria have circular DNA, and same thing with chloroplasts, and so it's T. 4. Which statements about viruses are correct? They contain DNA or RNA. Yes, they are. There are retroviruses or adenoviruses, and adenoviruses have DNA only. However, retroviruses have RNA only. So if they were to ask you, can a virus have the combination of both RNA and DNA? That is false. It can only have either DNA or RNA. They use host cells to synthesize virus proteins. That is correct. They have protease enzymes, but they cannot rely on them unless they are in a host cell, because the host cell is going to provide them with all the nutrients they want, and so that is right. They can have a protective coat of peptidoglycan. No, this is only for prokaryotic cells, 1 and 2 only, and so the answer is B. Moving on. What are features of prokaryotic cells, chloroplast, and mitochondria? Ribosomes smaller than 80S, correct. Their ribosome sizes are 70S. And circular DNA, that is true, surrounded by a double membrane. Then for bacterial cells, they're not surrounded by a double membrane. Only for chloroplasts and mitochondria. And so the answer is B. Students use Bendix solution to test a sample known to contain carbohydrates. At the end of the test, the solution was blue. So which carbohydrate could be present in the sample? So if the solution was blue, then obviously it does not have any reducing sugars, because blue indicates it's a negative test. And so the carbohydrate that's present in sugar should be non-reducing, like sucrose. Maltose is reducing, fructose is reducing, glucose is reducing. Moving on. Sugars with a ring structure can also have a linear structure. Which sugar molecules could be represented by a linear structure in the diagram? So obviously when we look at this linear structure, the first thing that should come to mind is glucose, but we have to count in order to make sure. 1, 2, 3, 4, 5, 6, obviously glucose, not any other monosaccharide. Not ribose, it's a pentose sugar. Not deoxyribose, it's only glucose. D. Glucose can also have ring structures, but it also has, as they mentioned here in the beginning, linear structure. Okay, which row matches each molecule to the type of bond that is present? An ester bond is found in lipids or in phospholipids, so we can cancel out amylase and glycerol. Hydrogen bonds are found in amylopectin. Actually, it's found in cellulose mainly. Amylopectin, not really, no. Disulfide bonds are found in antibodies, correct. It's not found in amylose because it's only found in proteins in order to maintain tertiary structures and quaternary structures. Now, just like in antibodies, it's needed to maintain the quaternary structure, which is when more than one peptide chain is found in a protein. Which row shows features of a carbohydrate polymer found inside animal cells? So, carbohydrate polymer inside animal cells, we have glycogen, and glycogen has both alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds, and it's actually highly branched in shape and more branched than amylopectin, which is like a compound that forms part of starch in plants, and so that is A. Now 11. The statements describe the structures of polysaccharides found in cell walls of certain plants. The polysaccharide is composed of 2 different monosaccharides. So for cellulose, first off, he said which statements are correct for cellulose. The polysaccharide is only composed of 1 monosaccharide, which is beta-glucose. The monosaccharides are joined by 1,4 glycosidic bonds, that is true. The polysaccharide contains pentose sugars. It's actually a hexose sugar, so it's D. The general formula for saturated fatty acids is CnH2nO2. Which of these fatty acids are unsaturated? Okay, so we have to count the number of carbon atoms and see if the number of hydrogen atoms is double it, then it is saturated, and hence the answer would not include it. So let's start with number 1, C10. So we have 10 plus 1 carbon atoms, that's 11, and a total of only 20, so that's like 19 plus 1. So that's a total of 20, when the double of 11 is 22, and so it's actually unsaturated. 1 is unsaturated. 15 plus 1, that is 16 carbon atoms, and then we should expect 32 carbon atoms, which we do have, and so that means 32. So this is actually saturated. Here we have 18 carbon atoms, and we should expect 36, but we're only seeing 32. So that is unsaturated. We have 19 carbon atoms, so we should expect 38, but we only have 33, so it also is unsaturated. So the answer is 1, 3, and 4, which is C. What enables triglycerides to perform their functions in living organisms? So triglycerides have hydrophobic and hydrophilic regions. Triglycerides do not have hydrophilic regions, only hydrophobic regions. They're talking here about phospholipids. Triglycerides have a high ratio of carbon to hydrogen bonds to carbon hydrogen bonds to carbon atoms. That is true. That's basically why they are energy rich and energy dense molecules. Hydrolysis of triglycerides releases metabolic water. Actually no, the hydrolysis, it's actually the respiration of triglycerides that releases metabolic water, and so it's 2 only, which is D. 14. The diagram shows examples of different bonds. Which bonds can hold the quaternary structure of proteins together? So quaternary, bond 3, and bond 3 and 1. Because hydrogen bonds need to maintain tertiary structure and hence can also maintain the quaternary structure. So it's 1 and 3 only, which is C. Moving on. 15. Which row correctly describes the primary structure, secondary, tertiary, and quaternary structure of some proteins? Let's see. Primary structure determines the folding of the polypeptide. Well, actually, yes, it does determine the secondary, quaternary, and tertiary structure. And in tertiary structure, the protein is folded and coiled, and so I would say that is correct. Depends on hydrogen bonding between the side chains of amino acids. It's not between the side chains, it's between the CO group and basically the NH group, which is like not necessarily a side chain. It could be the parts that form the peptide bond itself. Okay, tertiary structure defines the overall shape and folding of the protein, and formed when two or more identical monomers, not necessarily identical, and so I would cancel it out because of both the first, secondary, and quaternary structure. Defines the order of amino acids in the polypeptide. That is correct. Usually forms immediately after polypeptide synthesis, not necessarily. Is held together by all types of bonding, because this is going to be formed during post-translation modification, which actually takes some time, so it's not necessarily immediately. Is held together by all types of bonds that occur in a protein. Well, not all of them. There's peptide bonds. It's not held by peptide bonds. Quaternary, so let's just cancel this option. C. Involves covalent bonds only. That is correct. Its peptide bond is a covalent bond. Secondary involves interactions between hydrogen and oxygen. That is true. Tertiary, essential for the function of enzymes and receptors. Correct, because enzymes are globular proteins. Being globular means it has tertiary structure, it is ball-shaped and is soluble. And same thing for receptors, they're also globular proteins. And so that is right. And quaternary structure formed when 2 or more peptide chains join together. Correct. So the answer must be C, but let's see D just to make sure. Involves peptide bonds between the side chains of amino acids. Obviously not. It's only between it's between the nitrogen and basically the carboxyl group and the amino group. It's not the side chains. Now for 16, which relationships could be investigated using a colorimeter? So the effect of light intensity on the rate at which the solution of a light sensitive dye changes from green to colorless. Sure, as long as there's color involved, then we can of course use the effect of light intensity on the rate at which the color changes from green to colorless. The effect of temperature on the rate of breakdown of cell membranes and tissues with pigmented cells such as beetroot. Alright, so if we understand what's happening here, the temperature is going to result in breaking down of the cell membranes, and so the pigment is going to diffuse to the surroundings. I suppose we could as well for number 16. Yeah, basically we can also do 2. Yeah, okay, the effect of pH on the rate of release of oxygen from the breakdown of hydrogen peroxide by catalase. This reaction does not have any color to it, so I would not understand how we can use color meters. It's actually colorless. Hydrogen peroxide is colorless. It releases water, which is colorless. The only thing that is released is gas, and we cannot use a colorimeter to measure the rate of release of gas or oxygen. The effect of light intensity on the rate of change of skin color of lizards that become paler in bright light. So, on the rate of change of skin color of lizards, how colorimeter works is basically you put the colored liquid inside a cuvette, which is like a small test tube, and then you place this cuvette inside the device, right? And basically, it's like some type of box, and it has these readings over here shown and some things that you can insert to it. So you cannot put a lizard inside that, right? You cannot. And so we cannot say this is right. It's B. Now which transport mechanism does not require concentration gradient to be present in order to take place? Okay, Fast diffusion and osmosis and transpiration all require a concentration gradient to be present. However, exocytosis does not. Now, for high concentrations of ethanol disrupt cell membranes by denaturing proteins and increasing separation between adjacent phospholipid molecules. Okay, as a result, a cell membrane can decrease thickness by up to 30% and become more permeable. Yeast cells release ethanol as a waste product of metabolism. In response to increased ethanol concentrations in their environment, yeast cells are able to increase the tolerance of their cell membranes to ethanol. Now, because ethanol is going to decrease in thickness up to 30%, I suppose it's going to need to become thicker and become less permeable. So which statement correctly explains a response to ethanol that could account for the increase in tolerance of yeast cell membranes to ethanol? So decreasing the ratio of saturated to unsaturated fatty acids within cell membranes helps prevent the tails of phospholipids on one side of the bilayer from sliding past the tails of the phospholipid on the other side of the bilayer. Okay, that's very interesting actually. If you decrease the ratio of saturated to unsaturated, that means you increase the ratio or the number of unsaturated fatty acids, which is really important for increasing thickness because they do not pack up against each other because they have these different shapes. So they're arranged farther away from each other, which increases the membrane thickness. And helps to prevent the tails of the phospholipids on one side of the bilayer from sliding past the tails of the phospholipid on the other side. Now that is actually how the phospholipids are going to maintain fluidity. If you are going to stop the phospholipids from sliding on past each other, then you're going to decrease the fluidity and hence you're going to decrease the permeability. And that is exactly a defense mechanism because ethanol increases permeability. And so the answer is A. Let's read the other options. Okay, increasing the proportion of palmitoleic 6-16 unsaturated fatty acid to oleic acid and C18 unsaturated also fatty acid and phospholipid of the bilayer increase the fluidity of the cell membrane. I don't think that's right because you're going to, yes, it does increase the fluidity, but because it's shorter, and obviously if you place shorter fatty acids, then obviously you're going to have a more fluid surface membrane, but that's not the point. We don't want it to be more fluid because if it's more fluid then it's going to be more permeable, and that's going to even make it worse because ethanol is already increasing the permeability. Besides the fact that you're increasing the proportion of unsaturated to unsaturated, which doesn't make sense because we're not trying to increase the unsaturated to unsaturated. It's mainly you're supposed to increase the unsaturated to saturated because then just like in A, you're going to help them not pack so close to each other, increasing the thickness and basically also not affecting the fluidity in that way. Activating a signaling pathway triggers the unfolded protein response in the endoplasmic reticulum. The unfolded protein response pauses protein synthesis and initiates cell death in yeast cells with a high proportion of misfolded proteins. Okay, this does not show that they're actually more tolerant to ethanol. This shows that they're actually dying because of ethanol. An increase in the proportion of ergosterol in the cell membranes prevents the accumulation of polar molecules such as ethanol within the cell. Ergosterol in yeast cells has a similar effect on membrane permeability as cholesterol in mammalian cells. So if you increase if you increase the proportion of ergosterol in cell membranes, prevents the accumulation of the polar molecules. Okay, increasing the portion of ergosterol in cell membrane prevents the accumulation of polar molecules such as ethanol within the cell. Well, first off, there's something wrong right here. Polar molecules, ethanol. Ethanol is not polar, and hence D is wrong. Let's move on. In an investigation, a plant cell was placed in pure water, and the initial rate at which water molecules entered the cell was greater than the rate at which water molecules left the cell. So that shows that the net movement of water is into the cell. And in the second investigation, a plant cell of the same type was placed in a solution with water potential equal to that of the cell contents. Interesting. So what will happen in the second investigation for a period of 5 minutes? Water will enter and leave the cell in equal amounts. Okay, it will not enter or leave because there will be net movement of water. There will be movement of water, but the net movement will be 0. And so water molecules will enter and leave the cell in equal amounts. Correct. Both at the initial rate that is less than R in the first investigation. I would assume so too because there is no steep osmosis or water potential gradient. So the answer is B. 20. The protein P53 is produced in a cell in response to DNA damage. This protein stops the cell cycle for a short period of time just before DNA is replicated so that the DNA can be repaired. So just before the DNA is replicated, that is not during S phase, because we know synthesis phase is when DNA is replicated, and what comes before S phase is G1, and hence the answer is B. And if you do not remember, this is the pie chart. So Don't forget this. Okay, so here we have G1, first growth period, S phase, followed by G2, and then we have mitosis, and then cytokinesis. Okay, now let's move on. Some parts of typical human chromosome are more numerous than others. Which parts are listed in order from the most numerous to the least numerous? From the most numerous will obviously be histone protein, followed by telomere, and then DNA molecule. Sure, let's read the other answers though. Centromere is only one. Of course not. This is totally wrong. Telomere, followed by centromere, followed by nucleotides. Actually, obviously not. He said from the most numerous to the least numerous, not the opposite way around, and so the answer is C. Which row correctly describes the stage of mitosis? The stage of mitosis in case of prophase, the nuclear envelope will disappear. That is correct. That's correct. Yes, it forms tiny vesicles that are not seen under the light microscope. Centromeres are going to replicate. Actually, no, this happens during G2. Okay, so let's see. This was wrong, so let's move on to metaphase. For metaphase, the nuclear envelope is not present. Correct. Centromeres will move towards the opposite poles of the spindle. Actually, no, they have moved during prophase. So that is wrong. Anaphase, the nuclear envelope begins to reform. Okay, sure, because it's a cycle. Centromeres split into two, and the spindle cell microtubules shorten, not some, all of them. So that's wrong. State of telophase, the nuclear envelope is going to reform. Correct. Says at 7 centimeters at maximum distance from the cell equator. That is true. Spindle microtubules break down. That is correct. So the answer for number 22 is D. 23. The diagram represents a nucleotide containing adenine. Which statements about this nucleotide are correct? The carbohydrate is a pentose sugar. That is correct because it's a nucleotide. Nucleotides have either deoxyribose or ribose sugar, which is basically they're both pentose sugars. The base contains nitrogen. It's a nitrogenous base. Of course it does. Base pairing occurs with uracil, possibly, or with thymine. It depends on what kind of nucleotide it is. Adenine is a pyrimidine. Actually, that's false. Adenine and guanine are purines. However, thymine, uracil, and cytosine are pyrimidines. 1, 2, and 3 is B. And if you're not familiar with what a pyrimidine is, it's basically a nitrogenous base that has only a single ring structure. However, if you look at a purine, it's larger in size and it's got a double ring structure. Okay, now let's move on. Which row is correct for the replication of a DNA molecule? Direction of DNA synthesis is going to be from 5' to 3'. Enzyme that adds complementary nucleotides to the lagging strand. So this is a common mistake actually. The enzyme that adds the complementary nucleotides is DNA polymerase. However, the enzyme that catalyzes the sealing of the sugar phosphate backbone, like with the Okazaki fragments and all on the lagging strand, then that should be the DNA ligase, because DNA ligase has basically this function. So on the lagging strand, there will be some fragments that are not complete, and DNA polymerase is going to be adding these fragments, but it's not going to seal the bonds between them. DNA ligase comes in and seals these bonds between the fragments, but not between the nucleotides, unlike DNA polymerase, and that's why the answer is C. Now, some students were asked to look at the photomicrograph of a cross-section of an unfamiliar material, describe what they could see. So the students described the cross-section of F as circular, that is correct. Hollow tube, you cannot see the tube, you can only see a circle. So I would say that's wrong. Okay, spherical, can you see a sphere? A sphere is a 3D shape and it has some shading on it, one only, that's C. Moving on. So the diagram shows relationships between phloem sieve tube elements, Xylem vessel elements, and companion cells. Which row correctly identifies what could be represented by numbers 1, 2, 3, 4, and 5? So 1 is a companion cell. Okay, 2 is the endoplasmic reticulum, and then 3 is the phloem sieve tube element, no nucleus, and Xylem vessel elements. That is correct, so the answer should be A, but let's see the other options. Companion cells, nucleus, there is no nucleus in the phloem sieve tube elements. Phloem sieve tube element, mitochondria, companion cell, nucleus, there's nothing in the Xylem vessel element except for a cell wall. The Xylem vessel element, no cytoplasm. Actually, there is cytoplasm in the phloem sieve tube elements. Why does an air bubble in a Xylem vessel element stop the flow of water? Because of the loss of cohesion. Just like that one question, we're going to ask about the fungal infection, and what would it stop? It would stop the flow in the aplastic pathway, it stops the cohesion between water molecules, and it stops the development of earth pressure. So mainly if you see anything going up the xylem and basically like an air bubble or like a fungus, and just basically it's to get stuck somewhere in the xylem, so when water comes all the way up here, it's gonna not be able to bond with each other anymore, and so that's basically why it would stop the flow of water, because of 2 only D. Which changes to the water potential and the volume of the solution in the flume-sieve tube element occur when amino acids are moved into a sink from the flume-sieve tube element? So let's draw this, of course. So this is a flume-sieve tube element, and they're going to be moved to a sink. Right, so what happens is that the water potential is going to increase, however, the volume of the solution in the sieve tube element is going to decrease. So the answer is A. 29. Okay, 29. Which feature describes of some xerophytic leaves reduces the rate of transpiration by decreasing the water potential gradient between the internal leaf surface and the atmosphere. So obviously, to decrease the water potential gradient is basically to maybe do something with the water itself, like basically increase the humidity, something like this. So for thick waxy cuticle does not do any of those. Leaves reduce the spines to reduce the surface area to volume ratio. Another wrong one. Stomata located in sunken pits, exactly, because when it's in sunken pits, then the accumulation of humid air is going to happen on top of the sunken pits, and so that's going to reduce the diffusion gradient for water vapor and hence reduce the rate of water loss. Elongated leaves that are swollen, storing large amounts of water, it doesn't reduce. Okay, another example of this would actually be when the leaves are rolled, and so basically they're going to trap this humid air. Another example is also when there's trichomes present, and these trichomes are going to trap humid air as well, so the answer is C. The diagram shows the cross-section through a mammalian heart, and which chambers of the heart are represented by G and H. So by G, so okay, let's look at the mammalian heart over here. So here, this is the left ventricular wall because it's extremely thick, and then here we have the right ventricle. So we can say the right ventricle is G, the left ventricle is, so the answer is D. Okay, so 31, blood entering the heart from the vena cava passed several structures before entering the lungs. 5 of these structures are listed. Arranging these 5 structures in correct order of blood flow from the vena cava to the lungs, which structure will be the third structure? So make sure to only list the structures that are part of the right and left ventricles, and not something that is present in the left side of the heart, because the vena cava delivers blood to the right side of the heart, however the pulmonary vein delivers the blood to the left side of the heart. So let's start with basically when it enters, immediately it's going to be facing the sinoatrial node, because this is just a patch of muscle just below the opening of the vena cava, and so basically the blood is going to first see the sinoatrial node, and then it's going to be going through the right atrium. So even if there was an option of the right atrium over here, the answer should be sinoatrial node and then right atrium. So sinoatrial node, that's 1. And then basically so since we don't have that, we're going to say the atrioventricular node is number 2. And then we're going to have not the seminal valve, left atrium is wrong, aorta is wrong, pulmonary vein wrong, and basically pulmonary artery is correct, but we're going to first pass through the Purkinje tissue, and then we're going to pass through the pulmonary artery, but before we pass the pulmonary artery we have 4 and then 5. So which will be third? Number 3 is the Purkinje tissue, the answer is B. 32. The bar charts show the quantity of endothelial, elastic, and muscle tissue in the walls of 3 blood vessels, P, Q, and R. So which row correctly identifies 3 of the blood vessels? So endothelial tissue is present everywhere. However, elastic tissue higher in P, so it shows it's an elastic artery. Muscle tissue is really high in Q, so it shows it's a muscular artery. In R, it has a low proportion of everything. I would assume it's a vein. So P, elastic, artery, muscular artery, and vein. Answer here will be A. The maximum pressure in each of the 4 chambers of the healthy human heart was recorded as during one cardiac cycle. The maximum pressure recorded was 3 millimeters mercury, 10 millimeters mercury, 25 millimeters mercury, and 120 millimeters mercury. So which value was recorded in the right ventricle? So obviously the highest value is in the left ventricle, because of it's going to have really thick walls with lots of muscle to contract and push blood to the entirety of the body. However, basically 3 millimeters mercury would be basically in the atria, right? Basically left and right atria. 25 would obviously be the right ventricle, so that should be C. Now, for number 34, the graph shows the pressure changes in 3 structures of the right side of the heart, and so basically during the cardiac cycle, and basically what representatives are represented by the line labeled T. So T is actually an artery, any artery. Pressure changes in the pulmonary artery. This is pressure changes in the ventricle, because as you can see, like during the time about like 0.4 seconds, yes, there's contraction of the ventricles, right? And basically the contraction of the ventricle, besides that the pressure is extremely high, that shows that it's in the ventricle. This is obviously the atria, right? Whatever type of artery it is. So T is the artery, right? So that's the pulmonary artery. Which statements help to explain why there is no cartilage in the walls of bronchioles? So cartilage would make the bronchioles too narrow, or gases must diffuse across the walls of bronchioles. That's wrong. Smooth muscle is sufficient to support the walls of the bronchioles, or the bronchiole walls do not need to change shape. Okay, but that's okay, the bronchiole walls do not need to change shape. Cartilage would not make the walls of the bronchioles too narrow. Actually, it would prevent the collapse of the bronchioles if it was there. However, bronchioles do not collapse. Actually, it does not need to be supported by anything other than muscle, and I think that's why the answer is C. Okay, 35. The photomark graph shows the cross-section of part of the gas exchange system of a mammal. What is shown by the line labeled V? So the line labeled V is the diameter of I suppose this is a bronchus because of but no, there's no cartilage here, and so I'd assume it's a bronchiole. 36 is B. 37. Which feature of the disease cholera decrease the spread of the pathogen Vibrio cholerae? The immunity of cholera after vaccination is short-lived, lasting less than 2 years after vaccination. 50% of people. This does not decrease the spread of cholera; it increases it. Up to 98% of people infected with Vibrio cholerae are symptomless carriers. Again, it increases the spread of cholera because if you do not know that the person in front of you has cholera, you would not really care if he provides you the food, and you would not really care to clean the food or apply any substances in order to prevent it from being, infectious to you and so on. Cholera rapidly kills up to 50% of people with symptoms that are if they're not treated. I suppose this does decrease the spread because if these people die, then obviously they cannot spread the pathogen. Okay, simple rehydration therapy successful treats about 99% of people with symptoms of cholera. This does not decrease the spread because 99% of people, that's not 100%, but that's basically only with symptoms. There's also symptomless carriers. And not everyone has simple rehydration therapy, so it does not really successfully decrease the spread of pathogen as much as the letter C. 38. Which row shows how penicillin kills bacteria? Process is inhibited by penicillin. The formation of peptidoglycan cross-links, correct. However, basically effect of bacteria. Water enters and bacteria burst. How penicillin is going to kill bacteria. The effect on the bacteria is that the water is going to enter and bacteria burst. Actually, that is correct. Yes, the water does not leave and bacteria dehydrate. That's wrong. 39. In the hybridoma method, what is grown by cell culture to produce monoclonal antibodies? What is grown by the cell culture is actually, the clones of the hybridoma cells, right? So let's see. Antigens, no antigens are grown, right? Okay, clones, that's what I said, but I said of hybridoma cells. Let's see the other options. Lymphocytes, we're not really growing lymphocytes, we're growing the lymphocytes which are cancerous and plasma cells fused together, so that's wrong. Myeloma cells, not just myeloma cells, we're growing a specific specific hybridoma cell that produces the monoclonal antibodies, and so it's B. Which type of immunity does a baby have at birth? So the baby is going to have passive natural, coming from, basically when he was in tertiary life and just a fetus, and basically the antibodies were passing to him from across the placenta. And also basically when the baby is born, obviously there's going to be breastfeeding, and so also antibodies are going to pass in the colostrum, which is basically the breast milk, and the first few days after birth it's going to be really rich in antibodies, and so it's going to be passive natural. This is the end of this variant. I hope you found this video useful. If you have any comments on how I can possibly improve this video and the quality of this video, please write them down in the comments. Obviously, good luck everyone with your tests, and goodbye.